seminar was motivated by the request and need of local farmers in the region. So thank you for that. Soil-borne pathogens are increasingly a major concern and issue for organic vegetable growers, and the need to both understand the pathogen and understand the management strategies to be effective in the time and resources that you put into it is critical. So I put together this presentation specifically with that in mind, with the hope to give you the basics of the biology from to diagnosis, as well as a good understanding of the range of management options and how they can work on your farm. So I hope that this workshop paints that IPM picture of what effective soil-borne pest pathogen management can look like on your farm. The foundation for a lot of the people and science in this work today can be traced back to a single person, and that's Tom Gordon. So this presentation, this workshop today is dedicated to Tom Gordon. Tom was a researcher, professor at UC Davis in the Department of Pathology, and his research focused on plant pathogenic fungi. He was recognized nationally and internationally as an authority on diseases and was especially regarded in the Fusarium research community. And today's program is a great example of the legacy of his work. 4 of us here studied under Tom for our master's or doctoral degrees, and almost everyone here worked closely with Tom over the past 2 decades. Tom was really an extraordinary teacher, and he loved his students. And he had a signature course called Mushrooms, Molds, and Society, which eventually grew to over 500 students. And the high regard that students had for him was reflected in the applause that he would receive as he entered the lecture hall on the first day of class. He was really a legend for both the researchers and the students. He was beloved, and many spoke of his brilliance, creativity, humor, and his thoughtful, caring manner. So we miss Tom, and we're continuing his work here today. So with that, I'd like to start with our first presentation, and that will be from Dr. Cassandra Swett. Cassandra is our cooperative extension specialist located at UC Davis, and she is a specialist for plant pathology of vegetable crops. Thank you, Cassandra, and take it away.
And are you gonna stay visible? It always helps if I have like at least one person.
Sure, I can keep my video on.
If anybody else wants to turn the camera on, I don't know about the rest of the people, but I think it's a little hard. Hi, people. Oh, it's nice to see some humans. So, hi, I'm Cassandra Swett. Like Justine mentioned, oh, look, there's humans. I'm the CE specialist for vegetable and field crop pathology at Davis. I work closely with Margaret, as well as other small farm and organic farm advisors throughout the state, particularly for vegetables, which are, as I think many vegetable producers here can attest, are often heavily impacted by soil-borne diseases. So my program focuses predominantly on soil-borne diseases, and we work a lot on Fusarium wilt, and you'll see that we work a lot on tomatoes in particular. So a lot of my case studies will be coming from there. And I definitely have to acknowledge, following up on Margaret's dedication, that a lot of this presentation I actually built off of Tom's previous work, and I co-taught a class with Tom, and one of the lectures we gave was on this topic, and so I totally stole from his presentation for this. So his spirit was with us. Yes. Okay, so as we go along, please don't be shy about asking questions. I think it's easier to deal with things that are not clear kind of in situ. Okay, I will try to make an effort to pause as we go along, and you can put your questions in the chat. If you could keep an eye on the chat, because as a presenter it's hard to see it, that'd be really helpful. But then you can also raise your hand or just unmute and ask questions, and I think as a Zoom-based format particularly, it's a bit more engaging. Okay, so Fusarium wilt. Fusarium wilts are very diverse. but there are some sort of unifying features. I'm going to try to focus on those features for the majority of this talk, since Margaret wanted me to kind of cover general biology, but I'll be pulling from certain case studies like the tomato Fusarium wilt case study in particular for a lot of this. And hopefully you guys will get some management outputs, at least for that one particular disease from this. So what's Fusarium wilt? All right, well, generally there's unifying symptoms of all Fusarium wilt diseases. One is the vascular discoloration. You can see in the stem, and that's this browning of the stem in the corner there. And if you just cut into the plant, usually you can see that pretty clearly. And then another common trait is plant collapse or wilt, hence the name. And this can be very severe, high impact problem in many crops. We work a lot with Fusarium wilt and tomatoes, like I mentioned, and you can have field abandonment, significant reductions in yield to the point where there's just no point in harvesting it. And this is because wilts, plant collapse, it can, if it's the plant itself, like it's lettuce that you'd be eating, there goes your, your marketable product. Or even if it's not, say it's the fruit, then, there goes, the thing that was, say, protecting your fruit that maybe was preventing it from decaying or allowing it to fully mature. So there's a lot of ways that this plant collapse can influence yield outputs. Okay, so let's talk a little bit about the biology. So Fusarium, the Fusarium pathogen that causes wilt, which we'll talk about a little bit more in detail as we go along. It survives, it's a fungus, and it survives in the soil as spores. And it colonizes plant roots, okay? And from there, it goes into the xylem, which is the water conducting vessels in the plant, okay, like the pipe system, which often, if you do a cross section, usually you're gonna, depending on the plant, but you're gonna see that xylem towards the center. Okay, and the fungus will make spores in the plant, and they're carried up into through the xylem vessel. So we call that systemic colonization because it's through the system, okay? And that results in discoloration of the vascular tissue, that symptom that I showed you. And because the plant needs that water conducting system to move water, if you get clogging of that with fungal spores and it starts to die, then it can't transport the water, and then the plant wilts. And so that Fusarium wilt is really literally causing, like, basically a water stress-based wilt, where the plant is wilting because it can't successfully transport water. Okay. So in understanding Fusarium wilt, I think it's helpful to understand a little bit about this pathogen. And so this is caused by Fusarium oxysporum. And Fusarium oxysporum in general is common in cultivated soils around the world. It's everywhere, like everywhere. But for the most part, this fungus is not a pathogen. It just colonizes roots and causes no visible damage. So that doesn't really mesh with maybe what you might have seen when you had Fusarium wilt on your farm. Definitely pathogenic. What's going on here? So Fusarium oxysporums have diverse strains. And a very simple sort of explanation for that is that they very easily mutate, and they're very diverse. And so what can happen is, say you're a grower in Australia and you plant cotton for the first time, you have a very diverse population of these Fusarium oxysporums. And that might include a strain that could cause wilt in cotton. And it didn't evolve that way. It just so happened to exist. So it's a chance event. And that actually has been documented, particularly for Fusarium wilt of cotton. Australian researchers showed that it came from a native and highly diverse strain population. And then it just so happened to cause wilt in cotton. And now because this is so good at mutating, you end up with this huge diversity of crops that are affected by different randomly occurring strains that have emerged throughout the world. And we have over 120 host-specific strains for Fusarium oxysporum. And so when we talk about these strains, we call them special forms or forma specialis. And each one of those is specific to each crop. And this is really important to understand when we're thinking about crop rotation. Okay. So we have a Fusarium oxysporum FSP lactuci. Lactuci goes to lettuce. And then Fragaria goes to strawberry, not surprising. And Lycopersicum goes to tomato. And I won't keep going because there's like over 100 more, but you get the idea. And I often get this question from folks, okay, I have Fusarium wilt in my lettuce. I'm really worried about planting strawberry because I know strawberry gets Fusarium wilt, right? Or I've got it in my tomato. So am I at risk of getting Fusarium wilt in my cotton? Because I know cotton also gets Fusarium wilt, right? And so it's this question of, are you going to get disease in one crop because you have Fusarium wilt in the other, right? And so as I explained, you won't. Because each one of these forma specialis, like forma specialis lactuci, only cause wilt disease in a very small range of hosts, and it's going to be basically lettuce. Occasionally there'll be a little bit of an extension to a very closely related crop, but it's mostly very limited in terms of the crops it causes diseases in. So Fusarium wilt of tomato is different from Fusarium wilt of lettuce. They're different diseases. Okay. So any questions so far about our understanding of this pathogen, the diversity, and the symptoms that it causes before we move on?
You were saying it's basically a naturally occurring strain, a pathogenic strain?
Mm-hmm. Naturally occurring, randomly occurring strains that just by chance alone encounter a crop where they can systemically colonize and cause wilt. Yes.
In that it's naturally occurring, does it have any beneficial
Not that people have really found. And when you look for Fusarium wilts, there are incredibly rare events of Fusarium wilts in native crops. So like one example is the acacia koa in Hawaii, but mostly these are affecting these agronomic crops. And we think perhaps we bred out the natural, say, abilities for these crops, right, which would prevent wilt development in a native wild species.
Thank you.
Yeah. Any other questions that?
Dr. Swett, so is there any possible association where you have, say, Fusarium oxysporum lactuci in the field, and then with the similar condition, you tend to have, say, Fragaria, or is it other ways, like the other way around? It's just like is this so prevailing population that you have the lettuce from a specialty where you tend not to have the strawberry, especially from a specialty?
Yeah, that's a great question. Are the say environmental conditions favorable to one Fusarium wilt disease going to favor another Fusarium wilt disease? And this is important. The biology of these different strains are different, and that's going to be a prevailing theme throughout the rest of my little talk this morning. And like Kelley Paugh, who's here, who's going to be talking after me, she did a lot of wonderful work with Tom in the Fusarium wilt of lettuce system, where they found some very specific environmental conditions that were particularly favoring that disease. That don't really apply to say, strawberries and so on. So, there's going to be always a need to develop an understanding of the specific biology of each one of these, because they're going to be different in how they respond to the environment. That's a great question. Okay.
Actually, one more question in the chat.
Yeah, please.
If it's spreads by spore, can it be wind carried from one planting to another, or is it primarily via equipment? And just a little time check, you have about 8 minutes left. Yeah, so the spread is mostly through soil and equipment is our understanding. All right, so with the consideration of time, we're going to segue to crop rotation questions. So because each of these diseases is crop specific, crop rotation should be effective to manage Fusarium wilt diseases, right? But there's going to be some caveats here. So when we look at survival of this fungus in the soil, we know that it can survive for long periods of time, but there often is this question of how long is it going to survive in my soil? How long do I need to rotate out? Okay, so millions of spores infest the plant that gets diseased, right? And in addition, this fungus can produce survival structures, these chlamydospores, and they can persist in the soil predominantly within infected plant tissue. So when we look at survival, this is a work done by Mike Davis, my predecessor, where he looked at survival of cotton Fusarium, the cotton Fusarium wilt pathogen, and the lettuce Fusarium wilt pathogen over a 3-year period under fallow conditions. And while he found that there was a real clear drop in survival of the Fusarium wilt of lettuce pathogen, such that by 3 years it was pretty much safe to go back into lettuce, the cotton Fusarium wilt pathogen hadn't hardly declined at all. So in terms of the whys of this, when you think about the tissue being incorporated into the soil, we know that there's going to be variable decomposition rates of this sort of protective material. And this is work that Kelley did, where we found that there was variable decomposition rates of different crops. And we think that this really influences persistence. So it's not super helpful to just tell you guys that it's all different and depends. So to highlight some trends, there is definitely a trend for that 3-year time period as the minimal time period for decline of that inoculum load. This is Fusarium wilt of melon declining over a 3-year period. And this is Fusarium wilt of tomato declining to non-detectable levels within a 3-year period. And so over this 3-year period, what's happening is that in the absence of a susceptible host, that initially infested tissue is breaking down with our decomposers. We remove that organic material. This is predominantly happening during these warm, wet periods of time. And then we get, over about 3 years, ultimate propagule death. And propagule is just another way of saying parts of the fungus. Okay, so we have this general pattern of about 3 years as a good time to be out of tomato. Be out of, you can tell I talk about tomato a lot, be out of affected crops. But are all crops that you rotate to equally good rotations? So work that, again, Kelley and I did indicates no. So this is mirrored in work that Tom Gordon did as well with lettuce, as well as other crops, where we find that certain crops like cotton result in a lower level of Fusarium wilt when we planted tomato the following year compared to, well, obviously tomato, as well as crops like pepper. Okay, and the whys of this are nuanced, but one element that's important to highlight here is host range. So I talked about how we can grow these non-susceptible crops and that this results in attrition of those propagules. And that's true when you just have localized root infections like you can see here. But not all hosts are only infected locally. So this is a study that a group of us did looking at the ability for the Fusarium wilt pathogen to colonize plants systemically that were not tomato. And these plants were all asymptomatic.
And we did find that many of these with the asterisks had significantly lower systemic stem colonization than tomato. But there were several that were systemically colonized at similar levels to tomato. And we refer to these as typhoid Mary crops because they're asymptomatic. You can't tell that they are infected, but they are building up inoculum loads in the soil unbeknownst to us. So it allows the pathogen to accumulate. So some general patterns that we think we're seeing that are hopefully applicable to a wide range of the crops that folks here are growing. One is that the more closely related, if the crop is in the same, if say you're growing, you have tomato, and then you grow another solanaceous crop like pepper, and you can see here that seems to increase inoculum loads more than if you grow an unrelated crop. And this is work that Tom and Janess Scott did, showing that, again, unrelated crops are less infected than the lettuce. So that's one general trend, try not to grow in the same family. And then another has to do with grasses. So fun fact, there's no Fusarium wilts in grasses at all. They don't seem to support systemic infection. And when we look at the grass crops like corn and rice, they're the least colonized. So those are just a couple of patterns there. So any questions about survival in the soil, rotation out to other crops?
Cass, you have about 1 minute left, and you have one question in the chat about whether Fusarium can survive compost and whether that should be avoided if you're composting infected plants.
I'll touch on that in a sec. So when we think about crop rotations and other crops, or other hosts, we often think about other crops. But I did want to highlight that weeds can be important to management. So even if you're, say you're fallowing, it can be really important to think about managing your weeds during fallow. This is, again, some of the work that Kelley and I did where we had much higher Fusarium wilt levels in the weeds when we had weedy plots that were fallow in the winter versus plots that were not weedy. So these can be put together to create sort of a comprehensive crop rotation management plan, such as one based on the individual studies we do for each of these pathogenic strains. And so, for example, for the tomato system, we've been able to say that you should rotate out at least 3 years. You should avoid, say, cucurbits and sunflower and pepper. That grasses, as well as beans, which we didn't get to show you today, and cotton seem to be better. And weed management during fallow can reduce pathogen survival. And while there's some generalizations, it's quite important to think about this and study this individually for each wilt disease. So to address the question about other factors influencing survival, there's not just crop rotation, obviously. Solarization or steam to heat soil can help reduce inoculum loads. Compost, what work that Kelley and I did, found that compost actually seems to enhance the decomposition rates of plant tissue and may help in reducing Fusarium wilt inoculum loads. Anaerobic soil disinfestation is also an option. So these are nuanced systems, and we have a strong CE network, including our host today, Margaret, and myself, and many others. The Swett Lab, when we're not drinking beer, of course, is a core part of that. So, reaching out to that CE network to get help with your crop rotations is going to be, I think, an important component to helping you with managing your wilt diseases. And we've done this for many producers. So if there's additional questions, happy to take additional questions.
Our next presenter is Steve Koike, who is a well-renowned plant pathologist, and he is the director of TriCal Diagnostics. So thank you, Steve, for joining us today. We are very pleased to have your expertise. And we're seeing the moderation side, so you might want to switch your display.
Okay. Thank you, Margaret.
Yeah, that looks
How's
great.
that?
Great.
Good? Okay, great. Well, I'll move right into this because I know you have a very full agenda. And first, I want to thank Margaret for inviting me. It's a pleasure to be on your program, Margaret, and you have a lot of great information with a lot of great speakers. So thank you for inviting me. And Cassandra gave a great talk to begin with. I will build on a lot of the things she already mentioned, but my focus will be on diagnostics. I'd like to introduce the idea that diagnosing Fusarium, well, it's not necessarily easy. Sometimes things are very straightforward. Cassandra mentioned some of the main symptoms, but sometimes it's a little more complicated with this particular disease. And I'd like to start by asking Margaret a personal question. So Margaret, how confident are you on the care and the recommendations and the advice you're getting from your doctors and your dentists? Do you always believe 100% that their information for you, their diagnosis and recommendation, those things are always 100% spot on and perfect? Of course, we won't ask you to answer that. We could do that offline if you have some beef with your caregivers. But I'd like to introduce the idea that even in such an advanced science as medicine, it's not always straightforward. Do you always feel that 100% perfection is what you get from your caregivers? If not, are you satisfied with the 95% accuracy? How about 90%? Is 90% enough for you? And then unfortunately, we have our big COVID thing. It's a really good example of the complexity of diagnosis because the symptoms from COVID can overlap with other symptoms. That's a confounding and complicating factor. And then again, we have to be honest, somewhere in all of human medicine, there are places for assumptions and guesses. We don't have all the information all the time. And so sometimes the best diagnosis does involve some assumptions. So how comfortable are we with that? And what types of assumptions and guesses are acceptable and which ones are not? And then, as you know, in medicine, tremendous advances in the types of lab tests and clinical tests that could be run for us. But even these have limitations and there are issues with those. Again, because of time, we'll be quick here, but even COVID, right? Which test is the best one? And when do you take the test? Do you take the test at the 3-day mark of symptoms or 5 days? And then finally, I'd like to present the idea that even with excellent medical professionals, with excellent tests and technology, the results still have to be interpreted. Sometimes it's very straightforward. If that line is red and not blue, that's an interpretation that the test makes for you. Otherwise, a professional has to make a call here, yes or no. And that's just unfortunate, the limitations in human medicine. And again, if I was in person with you in the audience, I would challenge any of you to say that's not correct, but we won't do that because of the Zoom format. And real quickly, that was set up to place before you the idea that we have the same or similar situation in plant disease diagnosis. Sometimes we could be 100% sure about what we're seeing, but sometimes it's not as straightforward. Symptoms, sometimes confusing. We do have to make some guesses and assumptions at times. And in plant pathology, we also have some really excellent technology in terms of testing, but these are limitations as well, and they have to be interpreted. So to illustrate that, I want to introduce to you, Margaret, what I call the diagnostic certainty scale, okay? And I'm sure most of us in this field have some format of this in our minds, but sometimes you get a sample in from our customers, Margaret. We're sure, we know exactly what that is. There's no problem there. Other times though, we're not sure. And even if we run tests, we're not really, we don't have a really firm idea what might be going on because of those confounding factors. Just as an example, there's Margaret in her office. She's a very well-trained, very experienced professional. You bring in a pepper sample to her and she sees the symptoms. She already knows what this thing is. She sees the curled leaves, the plants defoliating, burned fruit. She's got this, right, Margaret? You got this. But she's going to go further. She's going to look for the signs. That sign means the actual presence of the organism. She sees the crusty white growth on leaves, primarily on the bottom side, not the top side. She's got this. She knows what it is. Then finally, there's a lab component. She can go to a lab, do some microscope work, and see very diagnostic structures. So she's 100% sure on this scale because this is powdery mildew on pepper, and she knows that. But on the other hand, you bring a sample to me of this garlic, and I'm in trouble here. I'm not really sure what's going on here because the symptoms pictured here can be characteristic of 3 or 4 different pathogens on alliums. And signs, meaning the presence of the fungus or presence of the pathogen, they're not clear. There's growth on there, but I'm not sure exactly what that is. And finally, we do lab work. And as a commercial lab, we do lab work on almost every sample that comes in, but we get a bunch of stuff in there. And so I'm really not sure what's going on here. And I'm really on that left side of the scale because of the uncertainties and the conflicting data or confounding data we're getting in this situation. So I'd like to present that as just an introduction to present to you the idea that Fusarium wilt, unless you really have perfect conditions and the perfect sample, Fusarium wilt has a broad, long range of certainty. Sometimes samples can come in and we're really sure what it is. On the other hand, we do not know exactly if it's Fusarium. There may be other things involved, multiple diseases. And so Fusarium wilt's more difficult to nail down. So I just want to present to you the idea that Fusarium wilt has this broad range of uncertainty or certainty compared to other things like powdery mildew, downy mildew, other diseases. And here's some reasons why we have that broad range and there are challenges regarding Fusarium wilt. First of all, the symptoms tend to be general. Cassandra did a good job of showing us what the typical symptoms are, but in some of these lists here, poor growth, stunting, collapse, etc., some of these symptoms can be caused by any number of other diseases. And so the symptoms are not necessarily diagnostic in itself. And that's linked to the next paragraph there. These symptoms are shared in common by other pathogens, especially other soilborne pathogens and other root pathogens. You have Pythium or Phytophthora causing stunting. It looks similar, at least to the general eye, of a Fusarium wilt disease. So there's some overlap there. And that's especially true with Verticillium. For tomato, lettuce, and other crops that get both diseases, sometimes Fusarium and Verticillium symptoms are identical or at least overlap enough to where it's not straightforward to diagnose it at the symptom level. And the vascular discoloration, which we've talked about already a couple times, very important to look for that for Fusarium wilt diseases. But remember that vascular discoloration can be caused by other pathogens and also physiological situations. There are plant stress and plant senescing scenarios that cause discoloration in the vascular tissue. And again, just superficially, looks very similar to Fusarium. So be careful about that. And then finally, we have challenges here for Fusarium because you may have Fusarium wilt in your plant, in your sample, in your crop, but we could also have other diseases in there. The other diseases may be very important to diagnose as well. And also the symptoms of the two diseases may cause some complications in terms of diagnosis. Just some examples real quickly. For example, in lettuce, there are fully 7 different pathogens on lettuce that cause some symptoms that could be considered overlapping with Fusarium wilt. So 6 other pathogens plus Fusarium, there's a little bit of overlap. Of course, if you are very experienced with lettuce, you're way ahead of us and you can make some early differentiations here, but just keep in mind there are other things that cause similar overlapping symptoms in lettuce. In strawberry, there are 3 pathogens, Macrophomina, Fusarium, and Phytophthora, that really look identical in some of the symptoms. I myself, I'm super familiar with these diseases. However, I will not field diagnosis with a grower because of these overlapping symptoms. It's too dangerous to be wrong, and that's not a good place to do a guess here. So that's symptoms and field aspects. We also have challenges in terms of diagnostics regarding the laboratory. So this is where people like Cassandra and myself and Margaret and Peter, we're involved not only with field diagnosis and careful assessment of symptoms, but we have a laboratory capability that we're going to use as well as part of the tools. So I'll be quick here because Cassandra did a nice job of covering some of this. We need to remember there are many, many species of Fusarium. There's a lot of them. And the species look very similar. Again, a professional like Cassandra and others, they can separate these quite easily, but it takes many years and a lot of experience to do that. and even experts get it wrong. So the many species can look very similar. And then as Cassandra pointed out, we have a lot of diversity and complexity in this group. And now we're looking specifically at the group that causes wilt, which is the Fusarium oxysporum species. That's the only one, I think, the main one that causes wilt on our agronomic crops. Within oxysporum, there's a lot of diversity, different strains and isolates. We have non-pathogenic ones that look just like the pathogenic ones. The pathogenic species do tend to be host specific, as pointed out earlier, but they can also infect non-host and act as non-pathogens. So we have a complicated picture here, a complicated scenario with oxysporum, which is the wilt pathogen that we're focusing on. I also want to acknowledge Tom. I had the pleasure of working with Tom as well. Here he is threatening me with a lettuce knife because he thought I was working too slowly. Not really, Margaret, you know Tom, but we do miss him a lot, and tremendous resource for all of us. Tom and I worked on this little schematic. This is kind of maybe a visual way of adding to what Cassandra pointed out quickly because of time. At the top there, number one, that number one circle, the soil has a lot of different Fusarium species, many, many species. One of those is oxysporum, as you can see in that green box below it. Moving to circle two, in general, we can separate the two the oxysporum group into two separate groups. So we have pathogens and non-pathogens. And in circle three, of the pathogenic ones, we have different strains or isolates that only go to one crop. So you have a Fusarium that goes to celery, and a Fusarium that goes to strawberry, and a Fusarium that goes to tomato. They don't go to other crops. They're very specific in their ability to cause disease on that crop. So this is a way kind of to schematically show how complicated this group is in terms of Fusarium and Fusarium oxysporum that are pathogenic to our crops. About to wrap up here, Margaret. One thing that's important, because of this complexity, it's good to remember when you're working with laboratories or extension people or commercial labs, whatever, good to remember that the complexity should be reflected in the reports. So if you get reports on a test, you should either read the report carefully and interpret it yourself and or follow up with the laboratory person to get further information. Here's some examples of how a laboratory report can come back to you. You'll say positive for Fusarium. So I hope today already, if you don't already were aware of this, I hope today you know that if you get a lab report that says positive for Fusarium, that may be all the lab can do, but it's not enough to really understand your situation with that sample. It may say Fusarium species. In some cases, that's enough. In many cases, it's not. We don't know exactly what species and is that important to your situation. So that's kind of a limited report if you just get Fusarium species. It goes on, Fusarium oxysporum, that's possibly a pathogen, but it could be a non-pathogen, as I pointed out to you earlier. If you get one that actually gives you the forma specialis, that's great information, but how did the laboratory derive that information? There's a lot of complexity to determine that specifically. So my suggestion is, in terms of our practice, we have very good confidence when we get Fusarium wilt on some crops, but we'll still be giving that footnote to the customer and saying, very likely this is Fusarium wilt, but unless you did some molecular work, actual plant inoculations, we can't know for sure, but presumptive this is probably Fusarium wilt given all the evidence we have at hand. And that's a limitation. We need to recognize that there's a limitation here. Just a couple more, Margaret, I'm done here. All of us want to become more certain in our diagnostic skills. So this is kind of for myself, and maybe other pathologists have a different way of looking at it, but I like to improve my certainty on Fusarium wilt diagnostics. And so I want to call this moving up the scale. There are considerations we can use here to become better and move up the scale in our certainty. And just quickly, a couple things. First of all, just keep in mind the crop you're working with. If you're working with celery, for example, only one disease really causes Fusarium wilt symptoms on celery. So you're much safer in knowing that's Fusarium wilt with the lab work than you are with a disease like lettuce, which there's 6 other things. If the symptoms are typical, that helps you. If there's a single pathogen, just one disease, that also helps you be more certain when you find a Fusarium in a sample. The cultivar, this is a good thing to just save yourself embarrassment. Just know which ones are resistant and which are susceptible. If you have a resistant sample that is known to be resistant to Fusarium and you get Fusarium, it could be a new strain, it could be a new race, or very likely that Fusarium is not a pathogen. So knowing the cultivar and susceptibility is also very useful. Working with what I call clean tissues are very important. If you're looking for Fusarium wilt, trying to avoid the roots, higher vascular tissue above ground is useful. And then of course, when you do get a Fusarium excisperum, we pathologists have tools to help you get more certain that's a wilt one. Does the morphology match, percent recovery? We have DNA technology to help us. And again, if you really need to know, you need to inoculate healthy plants. This is my last slide, Margaret, just to illustrate those points. If you're working with celery, upper right photo, that's the only thing that causes those symptoms on, a Fusarium is the only thing that causes those symptoms on celery. So that's a point of certainty when you get Fusarium. Bottom right, if you get high percent recovery from vascular tissue, that increases the certainty. If I take 10 pieces of tomato vascular tissue and get Fusarium excisperum type out of 10, that's better than 2. If you only get 2 out of 10, that's some uncertainty. Middle photo, that lettuce root, avoid roots, a lot of junk can get in there, including non-pathogenic Fusariums. If you can, not always possible, if you can, avoid isolating and working with samples that are compromised in that way. Instead, on that left, use stem tissue above the ground that has really great symptoms and is away from some saprophytes and non-pathogens. And the top left, again, we have some molecular tools and other techniques to help us further along the way to be more certain. This is my last slide for my pathology colleagues here. I had the privilege of attending Paul Nelson's Fusarium workshop at Penn State University. He's one of the early Fusarium gods in our field. And he was saying, well, if you discover a new species, here's some possible names you can use for that new species. So there's a Fusarium mygessii, Fusarium yorgessii, a Fusarium let's all gessii. And that's because, as he says, all little bananas look alike. All these Fusarium spores kind of look alike. So with that, Margaret, I again thank you and your audience for having me, and be glad to take questions probably later because of your time, but anytime people can contact us and we'd be glad to talk with them. Thank you
so much, Steve. Kelley Paugh is a postdoctoral scholar in Cassandra Swett's lab, and she is among those who is a graduate of Tom Gordon's lab and has done a lot of work with Fusarium. So she'll be covering a couple different topics today. So thank you for joining us, Kelley.
Another thing that we have looked at was how soil health management impacts survival of soil-borne plant pathogens. So it's another topic. So organic practices such as compost amendments and cover cropping are typically aimed at improving soil fertility. However, what's less well understood is how these practices affect the survival of soil-borne fungal pathogens. So plant pathogens can survive in soil in different ways. So for instance, some pathogens produce propagules that are then protected within plant tissue that's buried in soil, such that as the plant tissue decomposes, this leads to greater attrition of those propagules that were otherwise protected within that plant tissue. An example of this would be the Fusarium tomato pathogen, Fusarium excisperum homosocialis lactopersicii, which I'll hereon refer to as FOL. And this is different than, say, other pathogens which can persist in soil instead as naked propagules. An example of this would be sclerotia-forming fungi, such as the southern blight pathogen, Sclerotium rollsii. So sclerotia are these masses of fungal tissue which just are strong enough just to persist in soil, just essentially as naked propagules. An example of a picture of sclerotium is shown at the lower left there. It looks like a kind of a mustard seed for that particular species. And so with this in mind, so we could say that these soil-borne pathogens have different modes of survival. In the case of FOL, we could say that this has a cellulose-based survival structure, such that the survival structure is primarily plant tissue-based, and that how it persists in soil is tied to the degradation of plant tissue because it's protected within that plant tissue. And when that plant tissue degrades, the fungal propagules are released in the soil and it's going to degrade very fast. So that's why we consider it to be a cellulose-based or plant tissue-based survival structure. This is as opposed to, say, sclerotia-forming pathogens like Sclerotium rollesii, the southern blight pathogen. Or we could say this is the chitin-based survival structure in the sense that we have it's just a naked fungal propagule that's strong enough to sustain this soil as a naked propagule. So therefore, forces that would act on it would be those that primarily are those that degrade, say, chitin, which is the main structural component of fungi. So with this in mind, so we have, say, these two different, like, for instance, these two different modes of pathogen survival that we're talking about for these FOL versus the southern blight pathogen, Sclerotium rollesii. And with this in mind, so that came to the question of, like, how does soil health management practices affect these two different fungi, and does it differ depending on, like, their mode of survival? So in order to examine this, what we did is we looked at the effect of long-term organic management versus conventional management on the survival of both FOL and the southern blights and pathogen. And so to conduct this study, what we did is we took soil from the Century experiment at Russell Ranch at UC Davis. So the Century experiment is essentially what that is that for the last over 25 years, there has been field plots that have been managed either through conventional soil fertility management regimes versus organic soil fertility regimes. And these are both corn tomato rotations. It's just that with conventional, there's only synthetic fertilizers that are applied. Whereas with the organic, soil fertility is managed with the use of poultry manure compost amendments, as well as the use of a winter cover crop. So we took soil from these conventional organically managed plots at the Century experiment, and we put them in pots, and then we buried FOL-infested tomato residue as the survival structure for that pathogen, as well as southern blight sclerotia as the survival structure for Sclerotium rollesii. We buried it in these this soil from these different plots, and for this greenhouse experiment. And then we removed these pathogens after 1 and 2 months to see, to compare, in order to evaluate these different survival structures for persistence as a measurement of pathogen survival in these two different treatments. So this is showing results for FOL-infested crop residue that was buried in these two different soils of conventional versus organically managed soil in this greenhouse experiment. And so this graph here is showing residue weight loss percentage, which means is that, and so what this shows is that essentially the longer the bar, the more decomposed that residue is, which means there's decreased persistence of that survival structure in soil. So reduced FOL survival, the longer the bar, which is good. So interestingly, what we found is that FOL-infested tomato residue decomposed faster in organic soil than it did in conventional soil. So that's a good thing. So organic soil associated with reduced persistence of this Fusarium wilt pathogen. And just to further emphasize this, this is just showing residue that was FOL-infested residue in the conventional soil versus those that were recovered from organic soils. You can see greater breakdown of the residue in the organic soil versus conventional. So that's just kind of cool. You can see it, visually as well, not just based on the weight measurements. So this is showing results that we had for the southern blight pathogen, where we had that sclerotia, that chitin-based survival structure. And so for this, how we measured decomposition, or essentially persistence of the survival structure, was based on scoring for fragmentation. So of the sclerotia. So with a 1 being that the sclerotium was intact, it was not decomposed, it's just what you expect a sclerotium to look like. Whereas a higher number, like a 4, means the sclerotium is very decomposed. So higher the number, the more decomposed the sclerotium, meaning reduced persistence of this pathogen. So the longer the bar means that the more fragmented it is, lower survival. So we actually had a different result than what we found for the ethyl-infested tomato residue. In this case, Sclerotia decomposed slower in organic soil, which is not good. It actually decomposed faster in conventional compared to organic. So we actually saw a difference in how soil fertility management affects these two different pathogens. And it could be because they have different modes of survival. So overall, like as I mentioned, organic soil health management reduces persistence of seems like cellulose-based survival structures like the ethyl-infested tomato residue, but perhaps enhances persistence of a chitin-based structures like Sclerotia rollsi. This is perhaps a generalization that we can infer from this result. So with this in mind, okay, so this is not good for Sclerotia rollsi, but is there a way we can counteract this negative effect of organic management on chitin-based structures like the Sclerotia rollsi? So to look at this, what we did is we also added crab meal to these two different treatments of conventional and organic soil. And crab meal is primarily composed of chitin, which is the same element with the same, which is also what fungal tissue is primarily composed of. It's also chitin. So the idea behind adding crab meal to soil, which is a form of chitin, is that this could perhaps stimulate microbes to produce more chitin degrading enzymes, meaning that perhaps when we add chitin to soil, this could lead to more microbes that can degrade, like say fungal cell walls, and therefore leads to lower survival of say these chitin-based survival structures. And so this is showing the results for conventional soil compared to conventional soil with crab meal added. And we did not really see an appreciable difference in conventional soil when we added chitin or crab meal, chitin in the form of crab meal. But what was actually pretty exciting is that we did see enhanced decomposition when we added chitin to crab meal, chitin to organic soil. So where like say we did see a significant increase in decomposition of Sclerotia when you added crab meal to organic soil, such that it was now on par with what we saw for conventional. So that's really cool because it shows that when you add chitin to soil, it can counteract this effect, this otherwise negative effect of organic practices on southern blight persistence. So that's pretty cool. So you can still get the benefits of say organic soil fertility management practices, or if you're an organic producer, that's what you can use. So that's pretty neat to see what how adding chitin can impact that. So another thing that we looked at, just like as a side note, was that we also sterilized these two different soils to see how that also affected the difference in pathogen survival. So say like, okay, so we have sterilized conventional and sterilized organic soil. Do we still see these differences? And the answer is no. Like once you have these sterilized soils, there's no significant difference between these two different treatments on these persistence of ethyl-infested crop residue or the persistence of Sclerotia rollsi. There's no significant difference between these two different treatments once these soils are sterilized. And what this suggests is that there is a biological basis for this impact of soil health management practices on pathogen survival, such that the differences that we do see and how these soil fertility regimes affect pathogen survival could be due to, say, something like microbial communities, like the structure of microbial communities, the abundance of soil microbes. So this is something that would be inactivated in sterilized soil. So overall, in conclusion, what we found is that plant pathogens are differently affected by organic practices. And under organic management regimes, there was reduced persistence of a cellulose-based pathogen structure, such as wilt-infested residue, but we saw enhanced persistence of chitin-based structures like the southern blight Sclerotia. But as I mentioned, chitin amendment appeared to kind of counteract this effect, which is a good thing. And also we found, just looking at these sterilized versions of these soils, that there appears to be a biological basis for the effect of organic soil health management on pathogen survival structures. So that was a bit kind of hopefully not too complicated explanation for things. But anyways, thank you very much. And I look forward to also answering any questions related to this.
Welcome Peter Henry to the workshop today. He is coming from the USDA ARS, Ag Research Service, located on the Central Coast. And we're pleased to have you here. So thank you so much, Peter.
Yeah, thank you, Margaret. Can you hear me and see everything okay?
It all looks good and sounds good.
Okay, great. Thank you. Yeah, so a lot of really good topics today, and it's important to hear about all of them because really effective disease management strategies result from combining multiple different tactics. And the one that I'm going to talk to you about now is the use of Fusarium wilt resistant varieties, plants that are bred to not become diseased when Fusarium wilt pathogens are present in your field. And to do that, I will, okay. Let's see, is this working? Okay, there we go. So to do that, I will walk you through the names. We've heard about some of the names. One of the names that I'll focus on is the race structure that Steve brought up in his presentation and introduce the concept and how these names relate to each other. This will be very relevant to the next section of the talk where I'll describe the mechanisms of resistance to Fusarium wilt that are deployed most often by plant breeders. And this is called a gene for gene resistance. This is kind of going to get technical, but I think it's important and I think it's interesting to understand why these things work. And hopefully it will help you to apply this knowledge in the field in the selection of cultivars that actually have resistance to whatever pathogen is in your field. As we've already heard from other speakers, Fusarium oxystorum is often non-pathogenic, very common in soil as a common in soil and as a root endophyte. There are some strains that can cause disease, and they typically have a very narrow host range of a single or few crops. We give those strains this Forma specialis designation. That just means all the strains that are pathogenic to a single host. And I work most frequently on Fusarium wilt of strawberry. That is the crop that I have the most expertise on. And so a lot of the images and references in this talk will be about Fusarium wilt of strawberry. And that disease is caused by Fusarium oxysporum Forma specialis for Guerrier. The FSP stands for Forma specialis. It's Latin for special form. So that is the pathogen that causes Fusarium wilt of strawberry. And it is distinct from Fusarium oxysporum Forma specialis lycopersicii, which causes Fusarium wilt of tomato. And in addition to the Forma specialis designation, there are differences in cultivar resistances, and we find these meaningful to talk about. They have practical implications for disease management. So here you have two strains, strain 1 and strain 2. Strain 1 can cause disease on cultivar A, but not cultivar B, whereas strain 2 can cause disease on both cultivars. And these two strains would actually be called race 1 and race 2. That's what the race designation is referring to. And I understand things best in a visual way. I'm a visual learner, and I kind of see these names as a nested system where the Forma specialis is a subset of the species, and there are multiple races at times within the designation of the Forma specialis. And I promise you that us scientists, we didn't just come up with all of these names to, make it really confusing and try to have a good reason for us to be around as we're the only ones who understand these names. No, that's not the reason. It's because there is something of practical importance that is communicated by each of these naming systems. So biology is complex, and unfortunately, our names reflect this by necessity. So to understand this, let's try to understand this race designation, because it will impact how you can utilize host resistance, especially in a system like Fusarium wilt of tomato. Plant plants produce proteins that can recognize pathogens. And here, those proteins are depicted by the blue squiggles. And this is one of the most important ways that plants defend themselves, just recognizing when the pathogen is there to raise defenses. And each cultivar can have a slightly different array of these so-called guard proteins. And that allows it to recognize different pathogen threats. So here you can see the plant on the left only has 2 guard proteins, whereas the plant on the right has 3. And the Fusarium wilt pathogen will produce weapons when it's attacking the plant, when it's trying to colonize the plant tissues. And if the weapons have a different shape than the guard protein, the plant will not recognize them. And if unrecognized, that fungus can grow uncontrolled in the plant, systemically colonize it, as Cassandra mentioned, and cause disease. The plant on the right does have a guard protein that is the same shape as a pathogen weapon. And that means that it will recognize the fungus is there. And when that happens, the defenses are activated, pathogen growth is restricted, and the plant remains healthy. So the one functional difference in this interaction between cultivar A and B is this one guard protein. And most characterized resistance genes to Fusarium wilt pathogens of other crops function in this same way. For many decades, Fusarium wilt pathogens have been categorized into what are called races, shown here on the left, based on which weapons they have, and the plant varieties are characterized into based on their resistance genes that they have. And single gene resistance like this, where people have used genetics to unravel the mechanism, has been found in almost every single case. And in tomato breeding, there has been a long history of finding resistance genes and then observing the emergence of new pathogenic race. For example, the first strains of Fusarium wilt of tomato were observed a very long time ago in the late 1800s in England. Resistant varieties were not an option for any grower until after 1939, when resistance to race 1 was first discovered. In rapid succession, a race 2 strain and cultivars that were resistant to it were discovered in the 40s and 50s. And race 3 was first observed in North Africa in 1967 and then in the 80s in Australia. But it took time for it to become a problem in regions where commercial tomato production was happening. And race 3 resistance was identified also in the 80s. And today, a race 4 strain has not yet been documented, and race 3 resistance has become available in a wider array of commercially viable varieties. And it's widely suspected that at some point, a race 4 strain will emerge through random mutations. One of the reasons for this continuous emergence of new races is that you need both parts for this type of resistance to work: the pathogen weapon and the plant guard protein. If the fungus mutates, and this mutation makes it so that it no longer has the weapon that was recognized by the plant guards, it will typically still be able to cause disease. And this mutant strain would be considered a new race. It would be called a resistance breaking strain, because even though the resistance gene is there, it no longer provides protection to the plant. You can visualize this also with an interaction table like this. And you can see that the columns represent the cultivars, cultivar A, which doesn't have the resistance gene, and cultivar B that has the I gene for resistance. And the columns represent different races, where race 1 has this weapon that is recognized and race 2 does not. So even though the resistance gene is present there in cultivar B, because race 2 has lost the thing that it recognizes, it is no longer immune. So race 2 can cause disease on cultivar B with the I gene. And this is the current table of how FO-lycopersicide races interact with the I2, and I3 resistance genes. And this is important to understand because if you have a race 2 isolate in your field and you grow a race 1 resistant cultivar, hoping it will prevent losses to Fusarium wilt, there will be no help. It will be no help to you. So it's important to hopefully be aware of what race is in your field so that you can have a better idea of what varieties will protect you if you need to grow tomatoes in that same spot in the future. So in summary, resistance genes function by recognizing or detecting specific proteins made by the pathogen. Knowing the race that is in your field can help you select an appropriate resistant variety. And there's no inherent yield penalty for growing a resistant variety because it's really just this one gene, although the genetic background of that gene is brought into will have an influence on yield. So with this understanding of pathogen races, let's talk about disease control in your field. Unfortunately, your geographic region will not be very helpful for determining the race that is present in your field, as all races have been found across the state. Overall, it seems like races 1 and 2, there are some reports that suggest that races 1 and 2 are more common, but race 3 has been found across the Sacramento and San Joaquin valleys, and it cannot be ruled out by geography alone that it is present in your fields. There is a race determination test that can be conducted by a private lab. There are multiple labs in the state that will provide this service, but it can be costly. So it really depends on how important this is to you and your production system, whether or not you would want to pay for that service. A less expensive way that would need to be done in advance, a less expensive way would be to plant varieties that are indicator varieties with known resistance genes in your field, just a few plants every year, so that if Fusarium wilt does show up, you might be able to see what race it was based on their reaction. If you're interested in that technique, you could talk to a farm advisor who could recommend varieties to include, so that if you do get Fusarium wilt, you can look at their reaction to have an idea of what race is present. Another option is just to assume that race 3 is in your field, because most race 3 resistant varieties are also resistant to races 1 and 2. This could be limiting because there are fewer varieties that are race 3 resistant than are resistant to the other 2 races, but this would be a very simple way to proceed. Most seed suppliers do carry Fusarium resistant varieties. They usually mention this in the description of that variety. Also plant suppliers. Unfortunately, the race isn't always indicated. And if it isn't, you can try to contact the company or a farm advisor to see if they know, but it's sometimes not possible to determine. So it's also important to note that you can make a resistant plant by grafting a susceptible variety onto a resistant rootstock. And Margaret will talk about that, but it's a nice option if you have certain varieties that are susceptible, but they have really nice fruit characteristics that you like. If you can graft those onto a resistant rootstock, you may be able to continue production in a Fusarium wilt infested field. This is an example of what you might see on a seed catalog. Here, Beefmaster VFN is the variety, and the VFN stands for Verticillium, Fusarium, and nematode resistance. The race specificity is not known, or it's not mentioned, so you would have to search for that information elsewhere. Another example of this is with the variety Sakura, which is a nice sweet cherry variety that has high resistance to Fusarium wilt. but the race is not mentioned here. So you would need to contact the seed company or do some more digging to figure out what race it was effective against. And it's important to note as well that although VFN means Fusarium, F1 just means it's a hybrid in this case, not that it's resistant to Fusarium wilt race 1. So you probably realize that, but it's just important to differentiate that in the name of the variety. But sometimes you can find varieties that taste good and are resistant to Fusarium wilt races 1, 2, and 3. This is a variety that I personally really like, and the race resistance is known, and it's nice that it has such a wide spread of a wide package of disease resistances. So with that, I'd like to thank Margaret and Kelley for providing input and some photos for this presentation, and my funding sources and lab for supporting my work. And I can take some questions.
That was really great, Peter. Thank you. A really important piece of this puzzle is understanding further the biology and the evolution and how breeding plays a role as part of our IPM package.
Thank you.
Okay, so I will be talking specifically about grafting and the potential for grafting as a tool. And organic fresh market tomatoes are a really significant crop, and so I looked at this in both commercial fields and research fields to explore the potential of grafting. And even for diversified farms in California, many of them have tomatoes as a really sort of cornerstone crop, both in terms of high acreage and important income. And one of the constraints to good yields and good production are soil-borne pathogens. And at least in the Central Valley, and I'm more familiar with Yellow Solano, Sacramento County, the soil-borne pathogens are Fusarium, the Fusariums, a couple of them, Fusarium wilt and foot and root rot. We also have Verticillium wilt. We have southern blight. I haven't seen it on tomatoes much, but I know Cassandra gets tomato samples, and root knot nematode. And these are constraints for both organic and conventional growers because there are limited tools even for conventional growers to be managing these. And as Peter just described, a couple of these soil-borne pathogens, namely Fusarium wilt and Verticillium wilt, do have this major resistance conferred by resistance genes. And so this has been used by breeders to improve varieties to transfer that resistance to the choice varieties. However, especially market growers, specialty growers, they may have their choice varieties that do not have resistance to these pathogens. And so that really becomes a tool for grafting, where you have a variety that you really want to grow, but it does not have this resistance that you need. And I wanted to point out in the couple of examples that Peter just showed, the first one that showed Fusarium said high Fusarium resistance, and that was a hybrid. It did not list that it had Verticillium resistance. So it's important to realize that even a hybrid variety may not be always giving you the full package. So that's further importance for getting the diagnostics confirmed, and then checking back with these varieties that you really do have the resistance that you think that you're getting. And so this is, these are some grafted plants where you're taking the rootstock of an improved variety that has this resistance you're looking for and combining it with the scion, the above ground fruit producing part of your susceptible variety. And this is what a tray looks like. These are the clips that hold that graft union together. And when we look at the most common rootstock varieties that are available, largely we have two sort of seed companies producing these rootstocks. And the common ones are Estamino, Espartano, Fortamino, Maxifort, Multifort, Dro 138TX. And these are the major pathogens that they're looking for rootstock resistance. the Fusarium wilts, race 1, 2, and 3, Fusarium crown and root rot, Verticillium wilt, tomato mosaic virus. And if you look at these HR, that means high resistance, S means susceptible. So we have 2 examples of heirloom varieties susceptible to all these pathogens. And then we have good high resistance to all of those pathogens from these major rootstocks. When we move into a couple other pests or pathogens, root knot nematode, tomato spotted wilt virus, corky root rot, we see mixed efficacy from these rootstocks, intermediate resistance, susceptibility, high resistance. So, but really in terms of these soilborne pathogens we're talking about, we can get that resistance. Do note that southern blight is not a pathogen that we have resistance for through genetic resistance. So you're not going to get that from a rootstock. It is the rootstock specifically that has the resistance. And so in the case of tomatoes, we have strong adventitious roots. And so when I was doing these studies early on, we tried to do really shallow planting so that we did not bury the graft union. But as many of you know who have grown tomatoes, it's really unavoidable. Those roots are going to reach the ground even if they're not buried. The tomatoes tend to fall to one side or another, and those that stem touches the ground. So in my experience, planting them as usual is going to be the best course of practice, or the only practical source of practice. And it is true that those adventitious roots that are coming from the scion and growing into the soil do not have protection to the soil-borne pathogens. However, I think it's important to recognize that susceptibility is to some extent a numbers game. And so what I mean by that is that in order for an infection to become systemic, multiple steps have to happen. A specific root has to encounter the pathogen propagule in the soil. And so only a certain number of roots are going to actually encounter the pathogen. And then the pathogen has to have a successful infection. So there are hundreds of attempts that don't actually lead to successful infection. And so the more roots we can have that have resistance is going to increase our likelihood of not having an infection. And the smaller number of roots that are susceptible are still going to decrease our chances of getting an infection. So, you know, we're looking at this from an IPM standpoint, and having more roots that are susceptible is still going to reduce the risk of infection, even if some of those roots are susceptible. So I've been working with grafted tomatoes since 2017. Over the course of these 5 years, I've conducted about 14 trials, and these were on 10-foot long plots, 60-inch beds, 4 replications, single line planting of 12-inch spacing plants, generally just subsurface drip. And this is a combination of research fields and grower fields. And for the most part, these were not disease studies. So these were on fields that potentially had disease, but they were not disease field, and that was not a controlled part of the study. And this is what some of those field trials looked like. So I wanted to start quickly with this idea that one of the major issues of growing heirlooms is the annual yield variability. And so what I have here is a subset of the data for the non-grafted plants. And I wanted to just point out variability. So let's look at this first box plot, where you have about, let's say 12,000, it's about 12 tons to the acre yield. Well, that was in 2018. In 2021, this field, a neighboring field, got 24,000 or 24 tons to the acre. So that's twice as much yield between these 2 years. So the yield variation is tremendous. And for a crop that is high value and tends to be labor intensive, this can be a really tricky planning feature of growing heirlooms as a producer. So one of the questions was, could grafting actually help stabilize this variability that comes with growing heirlooms? And so this is the a subset of the data looking at 3 years. And we have Brandywine on top and Cherokee Purple on the bottom. And across the bottom are some of these rootstocks. And on the far right of each year, you can see the non-grafted yield. And so I'll walk through a couple, but if you just look at it, you'll see that basically there's a lot of variability. So if we start on the far left, on the top, this is Brandywine. The non-grafted is here, and you'll see some of those rootstocks had higher yields and some of the rootstocks had lower yields. The non-grafted has this AB on top, and that signifies the statistical significance. And because it has both letters, that means that it is no different than any of these other rootstocks. When we look here at the Cherokee Purple, we see the same thing. We see variability in increased yield or decreased yield depending on the rootstock. Now, if I had just done this in 1 year, I may have said, oh, okay, great. I'm going to go with Multifort and Maxifort and Dro 138TX. In both Brandywine and Cherokee Purple, those were higher yielding, though not significant, than the non-grafted. However, you then go to 2019, the next year, and you see the non-grafted here. Well, it just about flip-flopped. This year, these 3 varieties had higher yields, though not significant, than the non-grafted. So without getting further into this, over the 14 trials that I have done, this type of flip-flopping and variability between the non-grafted and grafted fruits occurred. And there, you know, some of the questions I had was, did specific rootstocks consistently yield higher than non-grafted, even if those yields weren't significant? What are their trends? The answer I have is no. Did the performance of rootstocks, whether they were higher or lower, correlate to the year, like whether it was a good year for tomatoes or a bad year for tomatoes? No. So this was disappointing, but it does point out the importance of repeating your research over time and space. So then I needed to ask this question in the disease context. And so I root dip inoculated the grafted and non-grafted tomatoes into the pathogen that causes Fusarium wilt race 1 and 2. And so this first plant that you're looking at is a grafted plant. You can see how vigorous it is. It's quite tall. It's robust. And I'm walking next to the non-grafted Brandywines that have been inoculated. And immediately you can see there's more light coming through. You can see exposed fruit. You can see the tops of the plants. They were much less robust plants and were impacted by that Fusarium inoculation. And now we're back to those grafted plants that were also inoculated with Fusarium. And so I just did this last year for the first time, and this was the data that I found. So we'll focus here on this control, which is the non-grafted that was inoculated with the pathogen. And all of these rootstocks were also inoculated with the pathogen. And you can see that in both the Brandywine on top and the Cherokee Purple on the bottom, all rootstocks had higher yields than the non-grafted. These yields are not statistically significantly higher, but they are higher in number. And one of them, or two of them, both Fortamino, interestingly, are significantly higher statistically than the non-grafted. So in the 14 trials that I did, this was the most compelling data that showed at least all of the varieties performed somewhat similarly compared to the non-grafted. So I'll be repeating this this summer in order to further understand how promising of a tool this is. Grafting in tomatoes was originally sort of pursued and developed with the loss of fumigants that were controlling soil-borne pathogens. So there is a long history of the success of rootstocks for managing soil-borne pathogens in tomatoes. However, we do know that there's a lot of specificity to the scions, the cropping system, location, environment, et cetera. So it does need to be tested in your region. But interestingly, another question to ask is, we know that diseases are not evenly spread in a field. So when you plant into a quote unquote diseased field, it is often still patchy. And so you are actually planting some of those grafted plants into non-infected or infested soil. So it is also important to note, well, are the grafted plants going to hurt my yield? And what we can see here is on the far right, we have non-grafted, non-inoculated. And you can see that performed higher yielding than the non-grafted, though not significant. But also these rootstocks were not lower yielding or significantly different than the non-grafted, non-inoculated. So an important nuance. So in conclusion, if grafting can give you as good of a yield as one grown without the pathogen in a field that is pathogenic, then that would be a win. The economics is the next question to look at. Would this be economically feasible? So a short back of the envelope cost, this is what it's looking like in California to graft tomato seedlings commercially. There are several nurseries doing it. The actual grafting is about 80 cents to $1. The rootstock seed is quite expensive. They're about 30 to 40 cents per seed. And they have to do an overseeding of the rootstock. So you have to add a little bit more. So it's about $1.10, $1.40 extra per grafted seedling. If we reduce, a little bit of that, let's say you're paying $40 for a 200 cell tray. On average, you're going to be paying about $1 more per plant to use grafted plants. So I hope that has been helpful in considering whether or not grafting is a tool that would work for you. And this was a lot of time and effort supported by the Specialty Crops Research Initiative and many people over those 5 years. Jesus Fernandez Bio joining us from Spain. It's about dinner time, so thank you for tuning in, Jesus, and we're really happy to have you here. I can't hear you if you're talking.
I was muted. Hi, everyone. Sorry, I'm going to share my screen.
This is the beauty of a Zoom meeting. Not just that everybody here is joining, all the viewers are from different parts of California, but also our presenters are from local and international locations. Thank you, Jesus. That looks good.
Can you see my screen?
Yes.
Okay. Good morning and good evening from here. Well, it's my pleasure to be here today sharing with you some of our research about biosolarization, which is an amazing sustainable tool to manage soil-borne pathogens using local byproducts. And as I said, it's very sustainable because we can take advantage of those waste that we don't, or byproducts that we don't want in our crops or in our food system, especially if they are high volume and low cost. The biosolarization is also a tool that is going to provide some benefits in the soil. It's been proven that it can improve the soil quality. And of course, it's a perfect tool to decrease the application of pesticides. And we want that to decrease the impact in the environment and also in the growers and in the populations that are next to the areas that we want to deal with fumigants or pesticides. Biosolarization is a practice that works. I like to show this study because it's a study that shows results comparing biosolarization against different fumigants, some of the most commonly used. And it's a study that has been measuring depletion of nematodes in the soil for 12 years and comparing results against these fumigants. And we can see that biosolarization is not the best, but it's among the best in controlling these nematodes. If we add the benefits, the additional benefits that we can have in our crops, then I think everything adds up. Another thing that I like to show is that biosolarization is a term that sometimes might be confusing because actually it mixes two practices that are well known. In the one side, we have solarization, and in the other hand, we have anaerobic soil disinfestation. So biosolarization is a kind of a combination of both management of practice management of soilborne pathogens. Just to briefly go through the process, how we apply biosolarization. The first thing that we need to do, of course, is to add a source of organic matter with organic carbon available to the microorganisms into the soil. And then we're going to irrigate that soil. Adding water is going to have two effects that I'm going to explain a little bit later now. And what we need to do is to cover this soil with a transparent film. It's important that it has to be transparent because what we are looking for is a greenhouse effect in the soil. So the solar radiation is going to hit especially the surface of the soil, and we can reach temperature above 140 Fahrenheit. And this is the first reason why we want to add water into the soil, is because the transfer of heat is promoted by the water. And then the other mechanisms that we have in the soil is that we have added some organic matter, and then that organic matter is going to be depleted by the microorganisms, and is going to create anaerobic conditions. And here is where the water has the second effect. Secondly, first is going to promote the microbial activity, and then is going to occupy the pores of the soil. So it's going to promote these anaerobic conditions that we are looking for. And why are we looking for these anaerobic conditions? Is because we want to ferment the organic matter that we have added, because this fermentation is going to produce some byproducts known as volatile fatty acids that are biopesticides. Among them, we can find some biological products like acetic acid, which is found in vinegar, propionic acid, which is responsible for our smell when we sweat, or butyric acid that also you can smell in raw butter. So all of these are like biological, biodegradable biopesticides that will easily dissipate from the soil after our treatment. So our general goal has been just to test a lot of different local byproducts to see how they improve biosolarization. And what we clearly observe is that the efficacy of the increase of the efficacy of biosolarization is going to be directly related to the biological activity of the organic matter. And by biological activity, I mean available organic carbon. For instance, if we have some compost or digestates that we are adding to the soil, those are very, the available organic carbon is very low because it's been already degraded by the microorganisms. So we are going to have a low impact in the efficacy. However, we can have other benefits by adding this organic matter to the soil. What we can do is to improve this biological activity by using low amendment rates of raw material, or you applying to the soil some compost or digestates that haven't been like completely mature. Or if we need to have a very strong treatment, what we can apply is a high rate of amendments that haven't been treated, that raw material like tomato bombs or almond hulls without any type of stabilization process. And I usually show this figure because it's very impressive. This figure that shows the mortality of a model wheat that we use in our studies is Brassica nigra, or black mustard. And in this study, where we apply this biosolarization with tomato bombs at 5% of the soil of amendment rate, and after 5 days of treatment, we observed that there was a complete depletion of the seeds in our experiment. And that's something that was not observed in the samples that were incubated at room temperature and or were solarized. So as I said, organic acids are the most, one of the most relevant compounds in this process. I like to show this figure because it shows that there is a very good correlation between the concentration of organic acids and the mortality of this model wilt. This figure shows the mortality at samples that were incubated at room temperature, meaning that we don't have an effect of the solar heating, and the feeding is quite good. However, if we do the same feeding in samples that were incubated under solarization, what we see is that the feeding is not as good as the figure above. And the reason is because in this case, we have two mechanisms inactivating the pathogen, in this case, the wilt. And still there's a significant correlation. And what we can highlight is that if at room temperature, we need around 1 milligram of organic acids per gram of soil to reach 50% of mortality. Under conditions of solarization or biosolarization, to reach this 50%, we need only 0.3 milligrams of organic acid per gram of soil. So now what I want to show is another different scenarios where we can apply biosolarization to control different soil-borne pathogens. The first scenario is in a tomato crop that for some reasons, market or other pathogens, we are not allowed to harvest, so we leave it in the field. The question is that, can we use this waste, this byproduct, to promote biosolarization? So we did a study where we grew tomato plants at two different growth stage, late vegetative stage and late fruit stage. And this is a control. We use the non-treated or solarized and then biosolarized with tomato pomace that we have seen that it works quite well. This is a picture of the tomato pomace. This picture shows how we integrated the crops or the amendments in the soils, and that's the experiment where we have samples that were covered with tarp, and then we have control samples that were not covered to avoid the effect of the temperature. For this study, we used 3 different weeds: common lamb's quarter, red root pigweed, and field bindweed. And I'm not going to show all these results. There are too much information here. I'm just going to highlight that the different seeds behave differently against biosolarization. For instance, if we see the results related to common lamb's quarter, we see that all the samples that were biosolarized, no matter what the amendment was, we have an increase of the mortality compared to the solarized samples or the samples that were incubated without the plastic tarp. However, if we look at the mortality of red root pigweed, what we see is that there is no effect of the solar radiation, but we see that the samples that were incubated with the tomato plants, they have higher mortality than the other 2 treatments. So this is an example of how different weed species can be managed differently. Another scenario is we have a orchard, in this case an almond orchard, we have almond hulls available to control a pathogen. For this study, what we focused was in the root lesion nematode, and we used as a byproducts 2 different varieties of hulls. The non-parallel variety, which is characterized by having a lot of hulls of this the green area that surround the green skin that surrounds the almonds, and it has more sugar, so it's more easily degradable. And then we have the pollinator variety, which is a mix between these hulls and the shells, so it's a little bit more harder to degrade in the soil. So we ran this huge field experiment. You can see the treatment. Each I think each row is around 900 feet, yards. And the treatments were controlled without any type of treatment, then solarized, and then biosolarized with the pollinator residues and with non-parite residues. So we went there and we measured the root lesion nematode in the soil before the treatment, and of course we found root lesion nematode in all the treatments. And the good news were that after any of the treatment, actually we depleted the nematode population in the soil very rapidly. Only if even after 9 days of treatment, even if we kept the plastic in the soil for 41 days. Only if we zoom a little bit on the results, we can see that there is some trace nematodes in the soils that were either solarized or biosolarized with the pollinator holes. Results were even more promising when we went there 30 months after the treatment, and we measured the relative abundance of the root lesion nematode, and we observed that it didn't recolonize the soil. The concentration with the relative abundance was still very low in our treatments compared to the untreated control. And the third scenario, which is more related to what we are talking about today, about Fusarium, is another scenario where we are in arid regions, like south of California or Arizona, where we had a lot of issues with Fusarium, and the space or the lettuce crops, they share space with these date crops. And the dates are producing this date paste as a byproduct, which is very promising because it's highly sugary. So it has a high, around 70% content of sugar, and that's exactly what we want because that's fermentable sugars that are going to produce the organic acids that we are looking for. So as for now, we are just testing at the lab scale how this paste can, if it can be promising or not. So this picture just shows how we test that in the lab. And I like to show because what we try is different amendment rates from 0, 0.26, 0.8, and 1.6, which the highest concentration is equivalent to around 6 tons per acre. And it's nice to see that after 8 days of incubation, there is a strong degradation of the date paste, and you can see that in the dark color that are observed in the tubes with the highest concentration. So what we did, we measured the organic acid concentration, and well, as we expected, there was a lot of accumulation. Actually, this is the highest concentration I have ever observed in my experiments, up to 7 milligrams per gram of total volatile fatty acids in the highest amendment rate. And if we zoom at and we look at correlation between the total of organic acids compared to the date paste, there is a significant positive correlation between concentration and accumulation of BFAs. And although we don't have still data on control of Fusarium, we think that this is really promising because in another study that I used anaerobic digestates. Digestates is the byproduct of anaerobic digestion that is used to produce biofuel. We observed that these treatments reached the concentration of 700, so 10 times lower the concentration that we have already observed in our previous experiment. And this experiment already showed a good impact in the control of Fusarium in the soil. So we are really excited to see how the results are going to be in this experiment we try in the field. So to wrap up, biosolarization is a very sustainable practice that has shown to be a good alternative to chemical fumigation, and it's more efficient than solarization. And the stability of the amendment is going to be key to improve efficacy. the more the concentration of the volatile acids that we get, the higher the control. It's also a sustainable tool to manage low cost, high volume byproducts. And I haven't shown anything today, but it also improves soil health. But it's not perfect, like everything. So we need to always be cautious. And sometimes if we apply a large amount of organic matter, may have some risks of phytotoxicity, of nutrients unbalance. And for now, we need a lot of application of plastic to keep these anaerobic conditions and the organic acids in the soil, similarly to what would happen with chemical fumigation. So I think the next level would be to develop some sustainable films, more degradable, or even some tests with using like water as a layer to substitute this plastic film. And with all that, thank you for your attention. You have my email here in case you need to contact me for any questions, and I'm happy to answer any question now.
Joji Muramoto. He's our Cooperative Extension Organic Production Specialist located in the Central Coast, Santa Cruz area. And so thank you for joining us, Joji, and the mic is yours.
Okay, can you hear me?
Yes.
Okay, thank you. Good morning, everyone. Lots of great talks, and I'm happy to be here. My name is Joji Muramoto. I'm a Cooperative Extension Organic Production Specialist based at UC Santa Cruz. I'm going to talk about anaerobic soil disinfestation, in short ASD, for managing Fusarium wilt and Verticillium wilt. So the outline of the talk, first, what is ASD? History, principles, effect, mechanism, and practices. Then talk about the effect of ASD on Verticillium wilt and Fusarium wilt. Use the strawberry as an example, the crop I've been working with mainly, but the practice is applicable to other crops as well. And finally, I'm going to briefly talk about integrated soil-borne disease management in organic systems. Okay, so this picture shows the outbreak of Verticillium wilt we experienced at UCSC Organic Farm in 2002. Our organic farm is diversified organic farms. We grow more than 40 crops per year. And regardless of 7-year rotation of strawberry, we had this outbreak because Verticillium, I'm going to talk more about later, Verticillium dahliae can be hosted by multiple crops, many crops. And it's hard to avoid the host crops such as lettuce and tomatoes. And so regardless of long rotation, we ended up having this outbreak. And our area summer is very mild compared to woodland here. Is it real summer? It's very cold. So we cannot do sterilization. And of course, we cannot use a fumigant in organic farm. So we're kind of stuck. But fortunately, we learned the method called biological soil disinfestation or reductive soil disinfestation, developed in Netherlands and Japan independently around year 2000, and then started to do experiment about it. And it worked for Verticillium dahliae. And the principle of this approach is acid fermentation in anaerobic soil. So basically integrating principle of sterilization and flooding for places where either practice is not effective or feasible. Santa Cruz is a great example. We don't have enough heat to do sterilization, and water is too expensive to do flooding, but ASD can work. So just as Jesus talked about it, ASD and biosolarization is pretty much the same. And basically ASD in sterilization feasible region is called biosolarization. ASD has 3 steps similar to biosolarization. First step is to incorporate organic material and feed the native soil microbes with carbon source. And the typical rate for coastal region is rice bran, using rice bran as standard material for us, 6 to 9 tons per acre. And cover with oxygen impermeable tarp to limit gas exchange and oxygen supply. And irrigate to saturation, not flooding, just saturate it first, and then maintain above field water, field capacity moisture content for 3 weeks. And then during the fermentation process, all the good stuff, organic acids and volatiles produce and suppress pathogens. The picture here shows the application in open field in coastal California strawberries, and also high tunnel system in Pennsylvania. Effect of ASBD has been demonstrated to multiple crops and multiple pathogens and pests. This is some example from the United States. California, as a carbon source, in our area, you use rice bran mainly, but the rice bran plus cover crop, or wheat middling, the coffee grounds, yellow crimson, other things, like Jesus mentioned. Labile carbon, available carbon, that's important. And we tested for strawberry and Brussels sprouts and some walnuts and almond tree fruits, tree crops as well. And Florida, they use molasses and composted broiler litter as a carbon source, and for vegetables and cut flowers. And Tennessee, they use crop residues, molasses, wheat bran for vegetables. And Washington, crop residue, plant residue, and rice bran for apple replant disease complex. For Fusarium oxysporum and Verticillium dariae, today's topic, many target crop have been treated successfully by ASD. So mechanism, again, Jesus mentioned already, but organic acid and some other volatiles and the microbial community shift and toxic ions work together to suppress pathogens during the fermentation process. And the strength of and the length and the strength of anaerobic condition is affected by temperature and water content, water conditions, and the type and rate of carbon source. This study monitored what's actually happening during ASD process in terms of metabolome, volatile production, and microbiome changes. And at the early stage, oxygen and sugar depletes, and CO2 and lactic acid increase. And then other organic acid increase, and towards the end, sulfides, very toxic pathogen suppressor, also being produced. And microbiome phase, first batches increase, but later decrease, and firmicutes and Clostridium increase towards the end. Not just level of anaerobicity, but the temperature is very important in ASD process. This is an example for Verticillium dariae control by ASD. X-axis is a strength of anaerobic condition. Higher the number, the stronger the anaerobic condition. And the Y-bar shows the number of microscotia Verticillium dariae in the soil. We compared two different temperature conditions, 77 degree Fahrenheit, block 1, and triangle is 59 degree Fahrenheit. As you see here, at higher temperature, 25 degrees Celsius, if the level of anaerobic condition exceeds this 50,000 threshold, most of the Verticillium dariae is suppressed. But that was not the case for lower temperature. This is related to a volatile production, such as organic acid and sulfides. At lower temperature, even though cumulative anaerobic condition is strong, those volatiles may not be produced sufficiently. And we came up with a threshold to control Verticillium and Fusarium oxysporum. Verticillium I just mentioned, in terms of cumulative EH, 50,000 millivolt hours or more, and temperature can be anywhere above 68 Fahrenheit. However, oxysporum, Fusarium oxysporum requires much stronger anaerobic condition and higher temperature. So susceptibility of pathogens varies. Susceptibility to ASD varies depending on pathogens. This is an example of Fusarium infested field ASD trial, untreated control, plants are dying. And then if you do ASD in the fall, disease is actually worse. But if you do it in the summer, much better. So to control Fusarium wilt with ASD, you have to do when temperature is high enough. And that is related to the character of the pathogen. Fusarium is saprophytic, which means that Fusarium can use dead organic material as a carbon source. They can grow using those carbon source, including rice bran. They love rice bran actually. So, and then at the lower temperature, they use it for themselves and multiply. So you have to do ASD when temperature is much higher using clear plastic. And Verticillium is not saprophytic, but no worry about it. And also this is a little bit side note, but Fusarium and Verticillium varies in the other important character. Fusarium wilt is hosted, Fusarium wilt fragaria is only hosted by strawberry, but Verticillium can be hosted by so many species, which is important implication for crop rotation. And ASD has can create a suppressive soil, which means it can prevent reinfestation of pathogen. This is an example of Verticillium dariae. And this data was taken 1 year after ASD treatment. After ASD, we grew strawberry for whole season, 1 year, and grew winter cover crop, and then grew lettuce, and then took soil sample. We still found a significant difference, ASD effect in Verticillium number in the soil. This seems to be related to the microbial community shift created by ASD. And this chart shows another experiment, but again, soil sample was taken 1 year after ASD treatment. And then the darker the color, bluer the color, the different the bacterial community. So ASD plots and the other plot, farrow or mustard cake plot, bacterial community is very different. And that seems to be related to soil suppressiveness created by ASD treatment. And this is another little bit sidetrack, but when you use lots of carbon source like rice bran, it releases nutrient as well. And this is the case of rice bran. How much nitrogen is going to be released from rice bran. And rice bran can be very good slow release organic fertilizer. And then for strawberries, by using rice bran 6 to 9 tons per acre, grower can reduce or almost eliminate pre-plant fertilizer. And it releases 25 to 30%, 20 to 30% of total nitrogen within 4 to 6 months. Lastly, I'm going to briefly talk about integrated soil-borne disease management in organic systems. ASD is not the silver bullet, and especially in organic system, it's very important to integrate other available tactics to control soil-borne diseases. For example, first step of soil-borne disease management is also, of course, it's disease identification. What pathogen you have in your field. Depends on the pathogen, you need to implement different tactics. That's the first step. And of course, you can use host resistant, use of resistant or tolerant varieties. That's important. And sanitation. So like there was a talk about this morning. Try not to spread the disease to one field to another. And then crop rotation. This is also a foundation of, could be foundation of soil-borne disease management. For Fusarium, typically at least 2 or 3 year break of the same crop. That's important. But Verticillium, avoiding host crop is very important. So what is a host crop from Verticillium? This is an example. Berries and artichoke and cucumber, watermelon, pumpkin, so many. But there are non-host crops as well. So I try not to include host crops during rotation process, at least for 2 or 3 years. And better yet, broccoli is known to reduce the Verticillium diet microscrotia in the soil by incorporating the broccoli residues. So that's my talk. Thank you very much for your attention. If you have any questions, feel free to ask.
Okay, our next presenter is Hung Doan, and he's one of our newest colleagues in cooperative extension. He is a small farms and specialty crops advisor for San Bernardino and Riverside counties. So welcome, Hung.
Thank you for the introduction, Margaret. Thank you for having me. I'm presenting on behalf of a bunch of other collaborators that work on me with this project, Tom being one of them, who's been a mentor to me as well. I'm happy that everyone presented about the devastating of fungal disease. So today I'm going to talk about one way you can manage it with effective hot water treatment. Basically, many vegetable seeds are prone to seed-borne diseases. Some plant pathogen can be in or on the seed. Some seed-borne disease of tomato include fungal pathogens such as Fusarium wilt, Verticillium wilt, as well as early blight, late blights. You have bacterial pathogens such as bacterial leaf spots, bacterial canker, bacterial specks, and you also have viral pathogen causing tomato mosaic virus, cucumber mosaic virus, and alfalfa mosaic virus. This is our Fusarium wilt. This is in the endosperms. If you can see endosperm of the tomato seed, this is on the seed coat. Everything that's glowing is the Fusarium that we made it to produce GFP that glow. Basically, contaminated seed can be an important first source of pathogen on the farm. So using clean seed is an important first step in the management of a plant disease. Hot water treatment can eradicate or at least reduce the level of pathogen in the vegetable seed. Hot water is used by commercial vegetable seed companies to basically treat the seed, as well with chlorine and with fungicide or bactericide. Hot water treatment are effective because it not only soaks the seed, but it also kills the seed on the inside as well without killing the seed. So the supplies you need for this is bag to hold the seeds. So any type of cotton bags, any nylon type of bag of stocking, cheesecloth, anything that's permeable to water. You need a water bath, preferably 2, one for the pre-treatment and one for the actual treatment itself. And you need thermometers and then screen for seed drying. So the steps in a seed treatment include harvesting of the seeds. So you want to harvest seed from healthy plants. And then the second step is pre-treatment. So you want to pre-treat the seed to prime the seed for the actual treatment. So pre-treat the seed at 100 to 110 Fahrenheit for 5 to 10 minutes. And then you treat the seed in 122 Fahrenheit for roughly around 15 to 30 minutes, depending on the seed. Then you cool the heated treated seed right away to prevent excessive heat exposure. Then you dry the seed immediately to avoid priming the seed, basically. So the whole process will cost you roughly $100 to $200. You could do it under $100 if you want. It's pretty doable for small farm and small scale. So this is a picture taken from genes. So you want to avoid taking seed from plants that look like this. So basically, this is Fusarium wilt. So visually inspect the field, inspect the plants before you collect the seed, basically. So this is Fusarium wilt. So you see the yellow flagging. So before you harvest seed, you want to look at the seed throughout the growing stage to make sure that it's pathogen free. So this one has Fusarium wilt. See the yellowing. And once you cut into the leaf petiole, you see the vascular discoloration inside as well as the fungus move up the plants. And when you harvest a tomato fruits, you cut the plants, you inspect the the vascular discoloration to make sure the pathogen is there. So if it's not pathogen free and you see that it's in this late stage of infection where the fungus have moved all the way up to the stem and to the fruits, you don't want to use those type of fruits. So you just want to discard that and try to look for a clean plant. So you also have Verticillium wilt. It has this V-shaped symptom on the leaves, and you see it in vascular discoloration as well. So when you see vascular discoloration and necrotic V-shaped lesion on the leaves, you kind of want to avoid those plants, harvesting fruit from those plants. So once you harvest the seed, this is seed that was harvested and then not fermented. So it's harvested dry, and then if you plate that seed out, natural infection is very low. So natural infection on a plant, even though the plant has symptom of Fusarium, the natural infection is low. This is maybe 1 seed out of 5,000 seeds is infected. When the seed is fermented, so a lot of the organic protocol to harvest the seed is basically ferment the seed. When you ferment it, that one seed with the Fusarium on the seed coat can multiply basically, and then most of your seed would be infected, roughly around 75% infection. And then when the seed is hot water treated, it basically eliminates the Fusarium on the seed in most of the case. So the material, so seeds, make sure your seeds are not pelleted. So pelleted seed for mechanical planting or seeding cannot be used. Prime seed or old seed should not be treated. Seed that exude gelatinous material, such as basil seed, cannot be treated. Seeds from cucurbits such as squash, watermelon, and then peas, bean, sweet corn, beets, and some other crop can be severely damaged by hot water. So certain seed to avoid. Pre-treatment of the seed. So basically first you loosely secure the seed in a cotton or nylon bag, like I mentioned. Then you pre-warm the seed. So basically you could run the seed under tap water for 5 to 10 minutes, warm tap water. So you put your tap water on warm. It's roughly around 108 to 120. That's usually the temperature, but usually 108 for about 5 to 10 minutes. This can basically warm up the seed, so that way you can put the seed in hot water treatment, and then it'll be ready. So the more seed you have, the longer you should pre-treat the seed for. So precision on this step is not crucial because you're not trying to eliminate the pathogen. Watch out for cross-contamination. So if you're using a hot water, a pre-treatment bath for one seed lot, you just want to dump it out or use a different one because if the fungus is in there, it will contaminate your other seed as well. So step 3 is the hot water. So you place pre-warmed seed into a hot water bath that will constantly hold the recommended temperature. So if you don't pre-treat your seed when you put into the hot water treatment, that can knock down the temperature of the water because the seed is cold, right? So if you pre-treat the seed, this will less likely to happen and it will prime the seed as well. So make sure you have 2 thermometer, 2 is recommended just to make sure the temperature is correct. So there's a number of hot water bath available. You could buy a scientific one from basically $55 to $1,000. The 55 is much smaller. You can do small seed batch. The more seed you have, the larger the water bath you will want because it buffer. The more water you have, it'll buffer the temperature, right? So when you add the seed, it won't fluctuate. Pre-treatment seed will also reduce this fluctuation, like I mentioned. And then a lot of the scientific water bath has some type of pump in there that can circulate the temperature to keep it constant. So if you're doing some like a $55 one, it might not have that. So you might want to get an aquatic pump, basically. A pump for a fish tank for like $5 to $10 can help you do that as well. A great one that we have all in our kitchen, this is our sous vide. I actually have some friends that do this with their seeds as well. So basically it holds temperature pretty well. It's now the price is much more affordable, $65 to $200. It has a thermometer in there, but just to be safe, you should put in another thermometer that's mercury or another scientific thermometer that can gauge the temperature. This thing also circulate the temperature, the water, so it's constant. The constant temperature is maintained basically, and it's pretty portable. So the right temperature matters. The crucial hot water treatment, the right temperature matters because the right temperature ensure that the seed-borne pathogen are eliminated and the seed will be viable. So you want to use a mercury thermometer or some type of waterproof scientific 2-probe thermometer. So always use more than one thermometer just to be sure. You don't want to kind of kill your whole seed batch, or you want the pathogen to be viable as well. So step 4, after you treat the seed, it's crucial to cool it down immediately. So you can put it into tap water and it will just, the heat will dissipate. If you don't do this step, the seed will continue to be warm, and that process will reduce the seed viability and also will probably kill some seed as well. The last step is to dry the seed. So before you dry the seed, you want to pre-dry the seed with a cloth towel. And then you spread the seed over a screen or a mesh and then store it at room temperature. So roughly about 85 Fahrenheit and below. If you have a fan around, that'd be great. If you don't, you can get a vegetable dehydrator and put it on to the fan setting. That works as well. So this is a hot water seed treatment protocol for a number of seeds. Basically, this is a reference on a growing work of other scientists who've done seed treatment. I primarily work with cotton and tomato seed, but this one is basically established for most of the crops. Tomato, you can actually go a little higher than 122. You can go up to basically 140 Fahrenheit, and they will still be fine. Always test the seed in small batch as well before you commit. With that, I'd be more than happy to take any question.
Thank you, Hung. That was a really fantastic presentation. We actually have 2 minutes. If anyone has a question they'd like to ask now, you can go ahead and unmute yourself.
Hey Hans, have you ever tried steam for cleaning the seed?
Try what? I'm sorry.
Steam.
Steam. No, I've never done steam. I've done chlorine and fungicide and other stuff. But steam can work, but then it's not uniformly, or it might heat it too much on the outside, not enough in the inside. So I'm not an expert at steam. But someone should look into that. Steve question is, are there data on how common seed-borne Fusarium is for tomato? We do have one in it's not published because I've been behind on a lot of things, but basically in commercial seed lot, it's very low because the seed are treated with acid, and that eliminate most of the seed-borne Fusarium on the seed coat, and also some on the endosperm. The one that's on the taproot and on the actual tissue itself is harder to remove. And when we did a seed lot, 10,000 commercial seed lot, you get 1 or 2 Fusarium. And of those, like 1 is pathogenic to Fusarium, to tomato. The rest of them are saprophytic Fusarium.
Thank you, Hung. And maybe
Thank you, everyone.
Steve. Oh, go ahead.
Thank you, everyone. And my email is there if you have any questions.
Dan is tuning in from Florida. He's the Director of Research and Development at Agricultural Solutions and has a long history working in California with soil-borne pathogens and biologically based management. So thank you, Dan, for taking some time to join us.
Thank you, Margaret, and thank you to the team at UC Cooperative Extension for putting together a wonderful program today. I certainly have been taking notes prolifically for the last couple hours, so I appreciate it very much. And I'm also grateful for the opportunity to share a little bit of things that I've learned along the way in managing soil-borne fungal diseases biologically. And so that's what I'm going to talk about today, biologically based approaches for the management of soil-borne diseases. And I'm going to use a case study for Verticillium wilt in the Salinas Valley. We'll start with the options that you can use for the management of soil-borne diseases. Soil disinfestation is probably the most widely practiced. And by soil disinfestation, you mean removing the pathogen from the soil or disinfesting the soil. And that can be either chemically using soil fumigants or other fungicides. It can be biologically, such as anaerobic soil disinfestation or solarization or biosolarization, or even tillage practices can be disruptive to pathogen populations and can be used to disinfest the soil at times. So soil disinfestation is inclusive, both biological and chemical and conventional. And I don't want to dismiss this practice because it is an important tool, because it can very quickly reduce the pathogen populations down to a level that they can be more easily managed biologically. Okay, so you never want to take that tool out of your tool chest. There are times when you have to go in and disinfest the soil to be able to start implementing biologically based programs. The second approach is soil augmentation, and that means putting beneficial bacteria that have been reared in laboratories or fermentation facilities and putting them back into the soil in an attempt to shift the microbial community to favor those that are known biocontrol organisms. For example, this is a commercial formulation containing 5 species of bacillus and a species of Bacillus. And this little 1 liter bottle is sold to growers. It's pretty common in different parts of the country. And this 1 little liter bottle is meant to treat 50 acres. The third approach is the manipulation of endemic soil microbiomes. Okay, and that is creating beneficial shifts through your management practices. And this can be cover crops or rotation crops, carbon-based inputs such as the use of rice bran or grape pumice or tomato residue, which our previous speakers talked about. In this case, you're putting carbon-based input either through the cover crop or another downstream waste product from another industry, such as rice bran, to create the beneficial shifts in the soil microbiome. There are many cases where naturally disease suppressive soils have been identified and characterized, and they include a range of different soil-borne diseases such as club-rooted cabbage, Fusarium wilt. The plant pathology field has identified soils that are naturally suppressive to Fusarium wilts of tomato, watermelon, and banana. Sugar beet cyst nematode, potato scab, the apple replant disease complex, TriCal certainly of wheat has been studied in great detail. Root knot nematode suppressive soils, and bacterial wilt of tomato. So we do know that in nature, disease suppressive soils do exist. Soil microbes suppress plant disease through multiple mechanisms, and this is an important thing to remember. There's more than one way that they suppress disease. One is the microbes directly compete for nutrients or colonization sites on the plant roots. 2, they can directly parasitize the plant pathogen. 3, and this is probably the most common, it's through antagonism, which is the microbes produce antibiotics or other enzymes that are detrimental to the survival or infectivity of the plant pathogen. And finally, these microbes can directly initiate a host plant immune response. So there are 4 different types of mechanisms responsible for disease suppressive soils, and in many cases, all 4 are involved at the same time. It's an important thing to remember is that soils contain a vast array of biological diversity. For example, in an acre furrow slice of soil, you can have 900 quadrillion bacteria, about 800 quadrillion actinomycetes, and 900 trillion fungi present in an acre furrow slice of soil. For those of you that are less mathematically inclined, such as myself, 1 quadrillion is equivalent to 1 million times 1 billion. Okay, so it's a lot of zeros. What that means is that there's an incredible amount of microbial diversity within an acre furrow slice of soil. Microbial interactions in soil are very, very complicated. Think of it as a bunch of networks nested within networks. So Each organism has an impact on other groups of organisms while they're also being impacted by similar organisms. So it is a cluster. It's not easily defined. And I don't think it's necessarily to tease out every type of interaction within the soil microbiome. What is important to know is that the effects of soil disturbance, either through tillage or fumigation or flooding, the effects of soil disturbance are temporary, okay? And the composition and the speed of microbial recolonization will determine whether a soil is conducive or suppressive to fungal plant pathogens and plant parasitic nematodes. Key points for creating disease suppressive soils. Beneficial microbes are always present in the soil, no matter what previous land management practices or the history of the land management practices are. Two, the changes in the soil microbiomes that favor beneficial microbes will be determined by the land management practices. And now I'm going to illustrate all this with an example that I've been involved with over the last 10 years in the Salinas and Paugh valleys of coastal California. Here we're focusing on Verticillium wilt, which within these two valleys causes over $100 million worth of loss every year, mostly on lettuce and berry crops. The principal disease management strategy for Verticillium wilt in the Salinas Valley for about the last 50 years has been to lease ground to a conventional strawberry so that he can fumigate the ground. And then when he's done growing strawberries, the lettuce growers come back in and plant lettuce till disease breaks out again. This is data from studies done by Krishna Subaru's lab in Salinas with UC Davis. And what Krishna's group did is they followed the density of Verticillium dahlia in the soil as measured by the number of microscotia per gram of soil, following a strawberry crop that had been fumigated with methyl bromide chloropicrin mixtures. So at the far left, this is after the strawberry crop was completed and the grower went in and planted his lettuce crop. The number of microscotia per gram of soil was less than 2. At the end of the lettuce crop, the microscotia density in the soil had increased to over 100. And 100 is kind of the holy grail for lettuce production. When you have more than 100 microscosia per gram of soil, you will see significant disease loss in the field. So the grower went back and planted a second lettuce crop. And at the end of that second lettuce crop, his microscosia count was almost 700 per gram of soil. And then the lettuce grower did what most lettuce growers do at that time. He went and found out a strawberry grower to lease the ground to, and he fumigated to bring the inoculum back down. So this cycle or yo-yo of reducing inoculum and then letting it build up by planting a septical crop has been occurring over the last 40, 50 years in Salinas Valley. So what a group of us did is we went to 2 farms that had a history of being fumigated with methyl bromide chloropicrin for over 40 years. And we went into the portions of these fields where they could not fumigate due to the proximity to a residential structure, the buffer zone. And we collected soil from underneath the plants after the Verticillium wilt ripped through that untreated area. The 2 locations were entirely different soil types in the Salinas Valley. One was from the Blanco district, which is where this picture was taken, and the other was on the east side of Salinas Valley in what they call the decomposed granite soils. This was a pretty large project, and it took place over a 5-year period and involved a group from the University of California, Davis, Department of Plant Pathology, and Driscoll's, and also Agricultural Solutions. This is characterizing the 2 soil types. The soil type A would be the decomposed granite, and you can see it had a pretty high sand component. Soil B, which was in the Blanco district. It is a more heavy clay silt soil with about 36% clay in it. So two totally different soil types. Yet both soils very conducive to verticillium wilt and had a history of soil fumigation since dating back to the 1970s. Okay, we took two disease management practices that had demonstrated success against verticillium and other fungal plant pathogens. One was broccoli rotations, and most of that work has been done by a group from UC Davis. And broccoli rotations have been shown to reduce the incidence of verticillium wilt. We're
Sorry,
also looking
interrupting,
at two different groups. One,
Did other folks have
Yeah.
connectivity issues just then, or was that just me? Can anyone else chime in on that? I missed what you
Yep.
said.
Yeah, Margaret, this is Steve. I lost him for a minute or so.
Okay, yeah,
Okay.
if you could just
Yeah,
repeat.
There was a
the-
glitch.
Yeah, what you said on that last slide.
this soil
Uh,
type.
No, this one. I got about half of it.
Okay, broccoli rotations have been studied intensively in the last 10, 15 years in the Salinas Valley and shown to reduce the incidence of verticillium wilt. And crab meal as a source of chitin has been studied for both nematode and disease suppression dating back to the 1980s by independent groups from Auburn University and from Israel. So both of these different carbon sources have a history of being beneficial in the suppression of soil-borne disease. So we took both of these different carbon-based amendments and we looked at them individually and in combination using soil collected from the two fields in Salinas Valley with a history of verticillium wilt. We use eggplant as our susceptible host because it was easy to grow in the greenhouse and we could harvest the plants and quantify the vascular discoloration as an indication of disease. And we used a rating scale where disease severity is based on the amount of vascular discoloration. We also did a metagenomic analysis using the 16S ribosomal RNA genes and high throughput sequencing. So in layman's terms, we sequenced the soil microbiome to see which organisms were responsible if we got any disease reduction by the addition of these two amendments. The first thing we found, which has been documented, and certainly Cassandra has done a very good job in the past documenting this, is that microbial communities are very different based on the type of soil. All right, so soil type has a pronounced effect on the composition of soil microbial communities. So the red dots here are from soil type A, and the blue dots are from soil type B, and you can see the dots cluster based on the different soil type. But what was very encouraging is that within each soil type, the microbial communities, which are represented by the dots, clustered based on what amendments were added to the soil. So here's soil type A, here's soil type B. The orange dots are control treatment. The green dots are the chitin amendments. The red dots are the broccoli amendments. And the blue dots are where we combine broccoli and chitin. So even though the microbial communities were different in these two soil types, the effect of the two selective carbon sources were the same. Then we looked at the disease score based on community structure. And what we found is that the amendments individually had some effect on suppression of disease, but combined amendments had a significant effect on suppression of disease. To read this graph, we look at the disease score on the y-axis, and everything above a 2 means that you have significant vascular discoloration, and it correlates to a significant amount of disease in the field. So the orange dots are the microbial communities in our control treatments. All of them had significant disease. The green dots are the broccoli amendments alone. The pink dots are crab meal amendments alone, and the blue dots are the broccoli and the crab meal. So you get an individual effect from the carbon source, but combining the two, you get a synergistic effect in disease reduction. Then we went into the broccoli and chitin amended soils, and we teased out which bacterial genera were increased significantly by the addition of those two amendments. And then we went one step further and tried to identify if any of those genera whose populations were increased were documented biocontrol agents. And what we found is that 47 different genera of bacteria in these two Salinas Valley soils that are documented biocontrol agents or disease suppressive organisms whose populations were significantly increased when we added these two carbon sources. And what I really want you to take home from this slide right here is that of these 47 genera, only 4 are commercially available: Streptomyces, Pseudomonas, Bacillus, and Pennybacillus. That's not because they're the most effective. It's because they're the only ones that a commercial lab can grow in a fermentation tank, harvest the spores, dry them down, and guarantee you a shelf life of 18 months. So when you go buy your biocontrol agent and put it into the soil, just be aware that you're not necessarily buying the most effective biocontrol organism. You're buying the biocontrol organism that a company can grow commercially and make a business model from. So once this data was published, we went to the lettuce and strawberry industry and talked to the growers, and we got some initial participation. It was a little reluctant at times. I was fortunate I had the strong arm of Driscoll's to twist a few growers to get them to try this approach. And we lent our expertise to document changes in Verticillium populations over time when we started using this approach. Since then, it's become commercially adopted on a number of farms. About 5,000 acres in the Salinas Valley are using this approach. Some of the first growers that bought into this happens to be a very large lettuce grower down in Sprecklesville on the banks of the Salinas River. And he gave us a 39-acre section on the Pasco Ranch, Lot 14, Section B. And in 2016, he said, go ahead, you got 2 years to see if this concept works in my farm or ranch. So after his iceberg crop that had significant disease, we sampled for Mycosclerotia Verticillium dallii, and we had over 145 per gram of soil, and we know that over 100 leads to significant disease. We came in, we grew a broccoli crop with 300 pounds of crab meal, and then sampled Mycosclerotia at the end of the broccoli crop, and we got that reduction, but it wasn't a significant reduction, I think, to allow to grow disease commercially, grow lettuce commercially. So to compromise, what the grower did is he came in with a romaine crop using transplants. So we shortened the susceptibility window by growing transplants than rather from seed. Still continued the chitin amendments. And even though he grew a romaine crop, we got another reduction in disease in Mycosclerotia populations. And then by the second broccoli chitin rotation, we had reduced Mycosclerotia from an initial population of about 145 per gram of soil down to less than 10 per gram of soil. So this particular approach is working for Verticillium wilt in the Salinas and Paugh valleys. This is the actual sample grids that we used in this field. When we began the study, many of the sample locations had populations over 150 microscotia per gram of soil. Using GPS, we went back after every crop in the exact same location and sampled. And so by the end of 2 years, we had reduced inoculum to less than 10 microsclerotia in that same location where we had over 150. So the take-home messages here are beneficial microbes are present in all soils. Certainly, if we can come in to 2 different soil types in Salinas Valley that had been fumigated every other year with methylbromide chloropicrin since the 1970s and be able to create disease suppressive soils, that's a pretty good indication that your beneficials will still be present in your soil. If that's not enough to convince you, certainly knowing that you have over 900, what did I say, quadrillion bacteria per acre furrow slice of soil, you're not going to kill every beneficial microbe in that soil, just like you're not going to kill every pathogen, every pathogenic spore. So the ability of your beneficial microbes is going to be based on the land management practices that you adopt. And certainly, I want to emphasize something that Kelley brought up in her talk that I think is fascinating, very important to remember, when she showed that reductions in Fusarium wilt from the organic the organic rotation, but not in southern blight, Scrotium rolfsii, due to the type of microbes that are responsible for suppression of that pathogen. So by combining carbon sources that select for different groups of beneficial microbes, you increase the probability that you will create a disease suppressive soil. Another way to look at it is you mitigate the risk that you're going to have another outbreak of soil-borne disease. So biologically based management strategies, they should focus on regulating disease outbreaks using carbon-based inputs that are selective for antagonistic fungal pathogens, judicious selection of cover cropping sequences, and agronomic practices that promote plant health. And this third point, I can't emphasize enough. I spend most of my time now in Salinas Valley trying to correct water management or salinity issues just to create the opportunity to allow those soils to do the biological buffer needed to suppress Verticillium wilt. Without good agronomic practices, biological suppression of soil-borne disease or nematodes is going to be very, very difficult. That's it. Thanks, Margaret. I'll turn the screen back over to you.